In this class, we've explored the idea that chemical reactions that occur between some of our common functional groups have a really important influence on what chemical reactions occur in the body, and those chemical reactions that occur ultimately produce new compounds. A really important example we talked about was the reaction between two amino acids through a condensation reaction, right, to produce our all-important peptide bond, which is the foundation of the formation of polypeptide chains and proteins. Okay, but as it turns out, chemical reactions that occur in the backbone are not the only reactions that can happen within a polypeptide chain. The side chains of amino acids are also susceptible to chemical reactions. So in this video, we're going to explore a couple of these different modifications, a couple of these reactions that can happen. The first one is a reaction that's very similar to what we've already talked about. So some amino acids have the ability to get phosphorylated. So if we start with an alcohol on the amino acid side chain and we react it with a phosphate group. So here, let's say we have monohydrogen phosphate. Right? So in this reaction, we can see that we can attack our electrophile, and ultimately we will form a bond there and will lose water. So in this condensation reaction, what we end up forming is water and then a phosphorylated version of our amino acid side chain. So this is called a phosphorylation product. It's called a phosphorylation product or a phosphorylated amino acid because we've linked this phosphate group onto the amino acid. And as you can imagine, this can very much influence the chemical properties of any amino acid side chain that can get phosphorylated. So to figure out which side chains can be phosphorylated, we're looking for alcohol groups. Okay, if you go back and refer to your list of our common amino acids, the big ones are serine, threonine, and tyrosine. So these three amino acids are very susceptible to being phosphorylated because they all have reactive alcohols that can condense with a phosphate group. The other important modification we're going to think about in this class is the formation of disulfides. Okay, so the formation of a disulfide bond. Okay, so a disulfide bond is one where we have this linkage between two sulfurs. All right, and this compound can form if we react together two thiols in the presence of some oxygen. And this is an oxidation reaction that generates our disulfide. So in this case, we're looking for amino acids that have sulfur, and specifically a sulfur that has this hydrogen, so this thiol functional group. And the only one that we know of is cysteine. So cysteine, all right, this is our backbone. Cysteine looks an awful lot like serine, but it has this sulfur. So cysteine is the single amino acid that's susceptible to oxidation to form a disulfide. And when we form these disulfides, we very much influence the properties of our side chain. Not only that, but it actually creates this new covalent bond that occurs between our two side chains. And as we'll talk about in the next unit, this covalent linkage between side chains is the basis for some really important interactions that stabilize protein structure.